Well, my thanks to the organizers for the invitation to speak and to all of you for arriving so early this morning. What I'm going to focus on for this talk is the development arc of LentiGlobin for transfusion-dependent beta thalassemia. So this is a really exciting time, not just for Bluebird Bio, but for a number of companies that are now advancing the first wave of gene therapy drug products from R&D into the commercial arena. And to make that possible, we've been expanding our footprint. We have our first manufacturing facility in North Carolina, and we've built an organization in Europe to enable our first launch there. We have headquarters in Zug, Switzerland. So just a few words about transfusion-dependent thalassemia. It's a hereditary disorder that is, of course, marked by profound anemia. Patients require frequent blood transfusions for survival, and with those blood transfusions comes iron overload, and so they require chelation therapy to address the toxicity from that iron burden. The only potentially curative therapy today is allogeneic stem cell transplant, and the problem with that, of course, is that the ideal population are pediatric patients with a matched sibling donor, and most patients don't have that opportunity. And it's also burdened by the issues of immune incompatibility that come from receiving somebody else's stem cells and immune system. Beta thalassemia in the US and Europe is a rare disease, but it's much more prevalent in Southeast Asia. So the pathology underlying this disease is due to, let's see if I can get the pointer moving, yep, there we go, due to reduced or absent production of beta globin. This, of course, translates into a profound anemia. Whoops. And that results in the cascade of pathology leading to multi-organ morbidity and increased mortality in these patients. The outcomes with allogeneic stem cell transplant are generally quite good, but as I mentioned earlier, are usually restricted to pediatrics with a sibling matched donor, and even in that population, mortality can occur from the complications of immune incompatibility. And we see those consequences here with graft rejection, acute and chronic graft versus host disease as being the major toxicities, and infectious complications due to the immunosuppression that's required. So that leads to the obvious question, well, why gene addition therapy for TDT? Well, we know it's broken, it's the beta globin gene, and there's the potential, by using a patient's own stem cells, to have a similar efficacy outcome with a better safety profile by avoiding the issues of immune incompatibility. The foundation of the LentiGlobin program rests on the pioneering work of Philippe Lebouche, who developed the lentiviral vector system that would be able to successfully express the beta globin gene. This construct contains the beta globin locus control region, so you have a lineage-specific expression of the beta globin gene, and there's a very clever engineering feature in it, which is the threonine to glutamine amino acid substitution at position 87, and you'll hear me refer to this as T87Q by shorthand. And that takes a sequence found in fetal hemoglobin and introduces it into the normal beta globin gene, and that confers two very important properties, the anti-sickling properties of fetal hemoglobin, and it also allows us to precisely quantify the gene therapy product from a blood sample by just running it over HPLC. And you'll see how that plays out in the clinical data I'll present. So the work on the vector systems began way back in the 1990s. The first in vivo proof of concept work emerged at the beginning of the last decade. In 2002, this murine beta thalassemia model was published, in which the beta globin gene was expressed using a lentiviral vector system in hematopoietic stem cells and was able to normalize the pathology in that model. And here you see that reflected by the prevention of hypersplenism, which is classic in this model. Just prior to this, a murine model with T87Q modified beta globin was published in the sickle cell disease mouse model and showed similarly positive results. So that was a very exciting start that got us ready for the clinic. And then, so let's jump forward a couple of decades. Just last month, we received a positive opinion from the EMA CHMP for marketing authorization for LentiGlobin in adolescent and adult patients with transfusion-dependent thalassemia without a matched sibling donor. And here you see it referred to by the brand name Zynteglo. I'll refer to it as LentiGlobin in the presentation. All right, so what happened in those intervening years between the proof of concept animal data and the positive opinion? Let me walk you through the clinical data. So the first clinical proof of concept was published in 2010. This was a young man with E0 thalassemia. This is the type that's prevalent throughout Southeast Asia. And he was transplanted at Hôpital Necker by Marina Cavazzana and Philippe Labouche and their colleagues, and had a really remarkable outcome. And you see on the figure on the right side, on the y-axis, hemoglobin in grams per deciliter, months post-transplant on the x. And here we are designating each bar in the stacked bar graph as a different globin chain broken out by HPLC. So in the blue, that's the E variant of hemoglobin. The green is hemoglobin F, and this young man was making a large amount of hemoglobin F. And then in the red, we see the T87Q, so the gene therapy product. And I want you to note a couple of things. The kinetics of production of T87Q. So you'll see it took well over a year in this first patient to reach a maximum of about 3 grams per deciliter. Now, because he was making a fair amount of E and F, that was sufficient for him to stop blood transfusions about a year post-treatment. And that level of T87Q has been stable since. But this, of course, begged the question, could we do better? Because as you can imagine, if all we could produce was 3 grams per deciliter of the gene therapy product, that would prevent us from achieving transfusion independence in many patients with TDT. All right, so how do we do better? Well, we invested at Bluebird in improving the vector design, and as importantly, to introducing pharmaceutical grade manufacturing, so improving the potency and purity of the vector to improve outcomes. Here's an overview of how the clinical trials were run. So we collect hematopoietic stem cells using mobilization with GCSF and plerixafor. Those go off to a manufacturing facility where they're transduced and they undergo release testing. Once we know that the product is good, we can myeloablate the patient with busulfan, and that's full myeloablative doses of busulfan. The stem cells that are modified are then infused, and we follow the patient in the hospital for engraftment. And then after discharge, we'll follow these patients for 2 years within the treatment phase of the trial. And then all of our patients go into long-term follow-up, which is mandated by the regulatory authorities. And of course, typically what we'll do is we'll monitor these patients long-term for T87Q production, the hematology profile, and gene therapy specific markers like in vivo vector copy number. We'll look for insertional profiles by integration site analysis, and then safety. Here's an overview of the efficacy trials being run by Bluebird. The first was the 205 study, a single center study in Paris at Hôpital Necker. That's where the first transplant was done. The North Star trial, the 204 trial, is an international study in TDT patients. North Star 2 and North Star 3, also known as 207 and 212, are phase 3 trials in TDT. 207 focusing on what we call the non-00 genotypes. Those are in patients with the E and the beta plus genotypes where they make some endogenous globin. And then 212 is focusing on the 0-0 double knockouts and the patients with very severe beta plus mutations like IVS1-110 where they make virtually no endogenous globin. So it's a high bar for success. We've had a very collaborative and innovative engagement with the regulatory authorities both in Europe and in the US. We were among the first participants in a novel program in Europe called the Adaptive Pathways Program, in which we reached agreement with the EMA to pursue conditional approval for LentiGlobin in TDT on the basis of the 204 and 205 studies, and then interim data from the 207 study. The path we pursued in the US was for full approval on the basis of more mature data from the 207 pivotal trial, initially in the non-0-0 genotype patients, and then re-engagement with the regulatory authorities as additional data accrues in the pediatric population and in the 0-0 genotype patients in the 212 trial to expand the label. So where are we from the clinic at this point? As of the data cut that was presented at this past ASH, so September 2018, 40 patients with TDT had been treated with up to 45 months of follow-up. Let me focus first on the safety of LentiGlobin gene therapy. So the safety profile is really consistent with myeloablative conditioning with busulfan. And what I'll focus on for today is one of the toxicities that we see there, and that's vaso-occlusive disease of the liver. It's possible that in thalassemia, because we have patients who are iron overloaded, that the underlying liver damage may exacerbate that risk. And what we've also observed is that there may be some prolongation in time to platelet engraftment in these patients. So looking at engraftment first, we see here median neutrophil engraftment and platelet engraftment. All of our patients have successfully engrafted, but what you can see in the 204 trial and 207 trial is that some of the patients appear to have a prolongation of platelet engraftment. It is important to point out that in contrast to conventional stem cell transplant, our patient stem cells are manipulated in culture and are transduced with a lentiviral vector, so it's certainly possible that has some impact on platelet engraftment, but frankly the numbers are still too small to really fully characterize a potential effect there. And so as more data plays out, we'll have a better sense of that. With regard to veno-occlusive disease of the liver, we've seen 5 serious cases of VOD in the 204 and 207 trials. What's important to point out is that, there's a pointer, there we go, is that 4 out of the 5 of the patients received no VOD prophylaxis, and so as these results emerged, we instituted mandatory VOD prophylaxis in all of our transplant patients, and that appears to have had a beneficial effect on the risk of VOD. All right, so what about vector-related risks? So, of course, with retroviral gene therapy, we're all focused on the potential for insertional oncogenesis. Fortunately, knock on wood, across the field with all third-generation lentiviral vectors, we've seen no cases of insertional oncogenesis to date. We perform routine integration site analyses on all of our patients, and we've seen polyclonality so far without clonal dominance in the LentiGlobin program. We did see one case of myelodysplastic syndrome in a patient treated in the 206 sickle cell disease study with LentiGlobin treatment. However, we were able to isolate those blasts, and what we saw were classic cytogenetic abnormalities that can occur sporadically or have been described with alkylating agents like busulfan. And very importantly, we saw really negligible vector integration in those cells, so we know it's not being driven by the vector itself. The other concern with this platform is the potential for replication lentivirus, and of course no one has seen that with any of the third-generation vectors. We did see in the 204 study one patient who acquired wild-type HIV through conventional transmission routes and that was not considered related to the vector itself. And we sequenced the isolate, and it was a typical clade that you would find in that region. All right, let's dive down into the individual studies now. First, the 205 single-center study in France. 4 patients with TDT were treated. In this figure, what we see is each individual patient shown as a row, and the dark blue indicates the time from the last transfusion, months on the x-axis. And we see that all 4 of those patients were able to discontinue transfusions relatively quickly after transplant, and in fact, quite remarkably shown by those stars, 3 of the patients were able to begin therapeutic phlebotomy to reduce the iron overload that accompanies blood transfusions from thalassemia. And that's quite remarkable when you think these are patients who were dependent on blood transfusions for survival, in whom we're now withdrawing blood to lower their iron overload. So terrific outcome. Here we see that stacked bar graph format where the T87Q engineered globin is shown in red, the E-globin shown in green, and hemoglobin A in blue. And here we note, importantly, that all of these patients are making substantially more T87Q globin than that 3 grams per deciliter we saw in that first patient, showing the improvements that were introduced in the 205 and the 204 studies. Let's switch gears now to the 204 international study in TDT patients. Here's that same format showing time off transfusions, each row an individual patient. 8 out of 10 of the patients in this study with the non-00 genotypes were able to become free from blood transfusions. We see the duration off transfusions on the far right in the white embedded in those lines. And most importantly, we see that this is a durable treatment effect. So as you would expect if true long-term repopulating stem cells are transduced with the vector and engraft, this should last the life of the patient, as you would see with conventional transplant. We see on the far right the total hemoglobin at the last visit. And in these patients, we're achieving or even at normal, near normal or normal hemoglobin levels. Here we see the total hemoglobin and the T87Q median values over time, and focusing on the red, which is the median T87Q, we see we're about the 5 to 6 gram per deciliter range, stable over time, and so again, as we saw in the 205 study, substantially more than we saw in that first patient. But that, of course, begged the question, could we do even better so that we would feel quite confident in achieving near normal or normal levels of total hemoglobin in as many patients as possible, and feel confident we could actually tackle patients with the 00 beta thalassemia and achieve transfusion independence in those patients as well. So one of the observations in the 204 and 205 studies, not surprisingly, was that the drug product vector copy number correlates nicely with the in vivo vector copy number, and that of course is associated with the amount of T87Q globin that we're expressing. So our scientists devoted quite a lot of effort to screening large numbers of compounds that might enhance transduction, one of whom is sitting in the back right there. Yes? And so what we did is we applied what we learned from that work to a refined manufacturing process in the 207 and 212 trials, where we introduced transduction enhancers into that process. And so how did that play out? Well, we see that right here, where we're comparing the 204 and the 207 drug product characteristics. On the left panel, vector copy number of the drug product, and here we see on the right the proportion of CD34 cells transduced with the vector. And really a dramatic improvement in DPVCN and proportion of cells transduced. So that was really gratifying to see. And then the next question, of course, is how did that play out in the clinic? Here we see a figure showing the time off of transfusions for these patients. In the first 11 patients in the 207 trial, we were able to get 10 of the 11 off of transfusions. The second patient, second from the top, had a less favorable outcome and had to resume transfusions. But we saw a very encouraging outcome in the rest. And here we see what's driving that. And so with the exception of that second patient from the left, we see large amounts of T87Q being produced in the neighborhood of 9 or even more grams per deciliter in many of these patients. You see the total hemoglobins at the top, with most patients achieving near normal levels. You see the patient's genotype down below and the month post-transplant in the blue. And you can see that these levels of T87Q are rising very rapidly. And again, in contrast to what we saw with that very first patient. So typically in the first 6 to 9 months of therapy, we're seeing a near plateau of the T87Q production, though we're able to see it continue to rise in many patients even beyond that. And importantly here, as we plot that out over time, the T87Q in red and the total hemoglobin in blue, we see that treatment effect is stable with time. And this is really exciting. So this is the bone marrow histology from the first patient treated. And here we see a really unhappy looking bone marrow at screening. And then 12 months after gene therapy, a really dramatic normalization of the bone marrow histology. And on the far right, you see actually the myeloid erythroid ratio is normalized in these patients. All right, so what's next? The 212 study, North Star 3, is still underway. This is focusing on the genotypes in which patients make very little or no endogenous beta globin. At the time of ASH, when we last presented the data, we had data on 3 patients infused, only one of whom really had a reasonable amount of follow-up. Here, this patient in the middle, who with the HPLC data we see at month 9 was making over 11 grams per deciliter of T87Q, had a total hemoglobin of 12.8, so almost normal. And then we had at month 12 just a total hemoglobin level, which is normal, 13.8, with no blood transfusions in the prior year. So very encouraging data in the 00 genotype patients, showing how the improvements in manufacturing played out. And then finally, just one word about sickle cell disease. LentiGlobin is also being studied in patients with severe sickle cell disease. We recently amended the 206 trial to expand out Group C, in which we're introducing the refined manufacturing process. And we also have introduced a novel composite primary endpoint based on T87Q globin and total hemoglobin. We plan to initiate a pivotal trial this year, the 210 trial, focusing on a population with a high frequency of vaso-occlusive events at baseline and using the same novel composite endpoint. And so we look forward to presenting more data on all of these programs later this year. And finally, a big thank you to all of the study participants, to our investigators and study staff, and of course all of the Bluebirds, past and present, who've made this work possible. Thank you. I think we have time for questions, but
we have 6.5 minutes for questions.
There's a mic in the middle, so maybe if people, well, is there a portable mic? Or you can holler. Correct. So the question was, well, the comment was the second patient in 207 had to go back on transfusions, correct? Yeah, so we did phenotyping analyses, and interestingly, we presented this data, I believe it was at EHA the middle of last year. We saw there was an lymphoid predominance in the drug product of that patient, which was uncharacteristic of the other patients we had treated. By inference, you could wonder whether that means that there were a fewer number of true long-term repopulating stem cells. Why there was a lymphoid predominance in that product, we don't know. Did the patient have a viral infection in the weeks preceding mobilization? Pure speculation, but this is something that we're continuing to evaluate very closely, applying high-resolution phenotyping tools like CyTOF to the stems, the cells that are going into the product, so we can try and figure that out. Good question.
Hi there, good morning.
Good morning.
Interesting to see lentivirus in this whole conference. Just curious, what was your rationale for not using AAV over lenti?
Well, there are quite a number of rationales for not using AAV and needing to use a retrovirus. So what we need is to modify the hematopoietic stem cells to have a durable therapeutic effect. Cells which are spinning off innumerable progeny, and so we really need to modify the DNA with a therapeutic gene, and AAV doesn't allow that. So you dilute the treatment effect if you used AAV, whereas with a lentivirus, we're able to allow the progeny to continue, the progenitors that spin out of these long-term repopulating stem cells to continue to act like drug factories and make the therapeutic protein in their progeny.
Thank you so much.
Yeah.
So we've been hearing here in the conference and other places, people pushing forward genome editing platforms, when a focus is important to maintain the regulation, the normal physiological regulation of the gene. And I see that in this work you maintain some of the regulation by putting the right factors on the vector.
Right.
Can you comment a little bit on the potential of lentiviral vectors to maintain physiological regulation in comparison to CRISPR or other genome editing techniques?
Well, so as you point out, the incorporation of the endogenous locus control region, the intent is to allow physiologic regulation of production of the engineered beta globin, so both lineage-specific regulation, but also feedback regulation. And so far, we haven't seen any patients making too much beta globin, hopefully because that feedback regulation is working. Certainly, one would hope that with well-executed genome editing, you're making use of the endogenous regulatory elements that are there. So if that's done, you don't really need to introduce those elements as you do with gene addition, but you can make use of the endogenous regulatory elements. In terms of whether that will be successful, we'll just need to let that play out in the clinical arena, but it's certainly very exciting.
Thanks.
Yeah.
Can you detail about platelet engraftment? The platelet engraftment.
Yes, so I can't provide any more details than I provided here. We, based on the small numbers we've seen so far, we've seen a few patients who have longer than anticipated platelet engraftment times. They all successfully engraft. And again, we just need larger numbers of treated patients to fully characterize the effect. And as I said, it's very possible it has something to do with how we manipulate these cells with both gene addition and with manipulation and culture versus standard transplant. Let me just make sure there aren't any other questions before I get back to you. All right, second round, I guess, if there's time for another question, or one more, yes. Yes, that is part of our regulatory strategy. So we've expanded the 206 trial, both to focus on patients who have a high frequency of vaso-occlusive events at baseline. That would presumably enable us to show a meaningful reduction pre-treatment to post-treatment, and also to allow us, hopefully, to show a correlation between what we call the globin response composite endpoint, so T87Q and total hemoglobin, with the kind of conventionally understood treatment outcome of reduction of events. All right, I think that's all the time I have. Thank you very much.